Se potete accomodarvi in modo tale che possiamo iniziare nel tempo previsto questa seconda sessione della mattinata. Se prendete posto, bene, buon pomeriggio, buona mattinata a tutti, benvenuti a questa seconda sessione della mattinata. Consentitemi, prima di iniziare appunto con le relazioni, di complimentarmi con Alessandro come Presidente e con tutto il Comitato Direttivo per il grosso lavoro che hanno fatto e che sono stati capaci di tradurre, diciamo, questo grosso lavoro in breve tempo in un convegno che si è rivelato, come è già stato detto diverse volte precedentemente, al di là di ogni aspettativa. Ma soprattutto mi sembra che una particolarità di questo convegno sia la presenza dei giovani. Ecco, io penso che i giovani rappresentino una garanzia che questa associazione possa andare avanti nel futuro. Bene, dopo aver fatte queste premesse, it's a pleasure for me to introduce Professor Alain Fischer, and my task is very easy, because I think that Alain Fischer does not need any introduction because he's known to everyone as one of the most important immunologists in the world. He's one of the pioneers of the primary immunodeficiency diseases, and his work, together with his group, has greatly contributed to the description of several forms of primary immunodeficiency and to the identification of different molecular mechanisms and genetic defects, which led to development of efficacious strategic therapy. And he put the basis of the gene therapy, just to mention the gene therapy for SCID-X1. And having him, together with Talal Shatila and Fabio Candotti as international speakers with us today in the first national congress of this new association, it's an honor and a pleasure. And I think that with these words, I'm sure to represent the thoughts and the feeling of the participants in this meeting, in this audience. Please, Alain. We are happy to listen to your talk on inborn error of immunity and vulnerability to EBV infection.
Thank you, Alessandro, for these very kind words, and thank you for the invitation. Thank you, Alessandro. This is really nice to be in Florence again. But before talking about EBV, I'd like to express my solidarity and hope your solidarity with our US colleagues. Today, they are demonstrating in the streets of most of the capitals of states in the US because of the unprecedented attack against science by Trump and his administration. So if you agree with me, can I suggest you to stand up just for a few seconds? Thank you very much. Okay. Thank you. Thank you, Talal. So I hope this is taken out of my time.
Oh, yes. You can start just from now.
No, okay.
30 minutes.
Okay. So let's talk about the Epstein-Barr virus. We all know about this virus that belongs to the herpes family. It's an encapsulated DNA double-strand virus with 100 genes has tropism for B cells, with the CD21 molecule being the receptor. He is the cause of, as you know, of infectious mononucleosis. But importantly, this virus has the capacity of persisting in some B cells, is in virtually everyone who has been infected with EBV, usually during the teenager time, but could be earlier or later. And very importantly, this is a transforming virus, and it is estimated that it is the cause, or at least a very strong predisposition factor for cancer, mostly lymphomas, but not only lymphomas, as you will see, in approximately 200,000 cases a year in the world. So it's, of course, this is important. And as you know, recently it has been shown, and this is very exciting, that EBV is associated with multiple sclerosis by mechanisms that are not yet fully understood. But I'm talking about Epstein-Barr virus and the way the virus is handled by the immune system, because, as I will then try to illustrate it, by studying inborn errors causing vulnerability to EBV, we may understand better how do we physiologically defend ourselves against the virus. So I remind you a little bit about the virus and its cycle. So the virus infects B cells, usually in the oropharynx. It leads actually in two steps to two cycles of the virus, the so-called lytic cycle, where all of the virus genes are expressed, leading to cell death and propagation of the virus, infection of other cells, but also in the so-called latency type 3, where only 8 genes of the virus are expressed. So the cells are not destroyed by the virus, and instead they are transformed and they can proliferate. So of course this is a challenge to the immune system, and of course there is a strong, and as I will further discuss, and as you know, there is a very, very strong response of the immune system with expansion, as you will see, of NK cells, T cells, and a strong antibody response. But some of the B cells that are infected with the virus will persist, and in a silent way, that is, no gene of EBV is expressed, so that these cells cannot be recognized by the immune system. And from time to time, for various reasons, the virus can, again, express some of these genes, including EMNA1 and others, in the latency type 3 mode, and again, generating a new immune response. And usually this is silent because the immune response is sufficient enough. Nevertheless, in some occasions, this may lead to lymphomas, especially with expression of EMNA1 and a few other EBV molecules, and or other cancers. So the challenge for the immune system is double. One, it has to prevent EBV dissemination, that is to fight the virus at the lytic stage. And second, it has to control the expansion of B cells or other cells that are infected with EBV. So it's, as I will discuss, this is quite a specific and serious challenge for our immune system. Just to remind you again about the immune response to EBV in terms of adaptive immune responses. On the left-hand side panels, taken from that great review published 10 years ago, you can see on the second and third panel, the expansion, the tremendous expansion of T cells, EBV-specific T cells that are found in the blood. This is why the disease is called infectious mononucleosis. And these cells can account for close to 50% of T cells, for instance, at a given stage. There is also an expansion of NK cells, and as shown on the right-hand side panel, of course, there is also the antibody response, you know, and that is used for diagnosis purpose. So the question I'd like to discuss now is, what are the critical effectors among all of these adaptive responses, plus, of course, innate immune responses against the virus? What is really critical to control the EBV? At what happens if it doesn't work properly? Well, there are 4 phenotypes that we know, and actually you will even see a 5th one, where EBV is not fully controlled and leads to disease, and disease that can be fatal. Of course, you know them. The most prevalent one is so-called B lymphoproliferative disorder, that could be polyclonal. So EBV-infected B cells do expand with a little control of them, and these may lead, in some occasions, to oligoclonal, monoclonal expansion with lymphomas. This is one phenotype. The second phenotype is HLH, hemophagocytic lymphohistiocytosis, that can be driven, as you know, by EBV. The third one is chronic active EBV infection, where the EBV is infecting T or NK cells and leading to an expansion and a very pro-inflammatory setting associated with the presence of EBV in usually monoclonal population of T cells or NK cells. And the fourth one, less frequent, but also found in immunodeficient patients, are tumors of smooth muscles where the EBV is present in smooth muscle cells, so-called SMTs. So when do we find these phenotypes? Or when we do not find them, I think it's interesting to look within the huge spectrum of inborn errors of immunity we know. The ones that are not associated with EBV, severe EBV infection, I think it helps to understand what is critical, what is not so critical. Interestingly enough, and maybe surprisingly enough, all of the so far described deficiencies in innate immunity, including, for instance, defect in response or production of type 1 interferon, does not lead to severe EBV, unlike what is the case for flu or COVID and so on. NK lymphocyte deficiencies, if they are selected, do not lead to severe EBV. Antibody deficiency does not lead to severe EBV, of course, provided the B cells are there. If not, this is not significant. And finally, or maybe even more surprisingly, immunodeficiencies associated with profound defects in CD4 T cells, so excluding CD8, like HLA class II deficiencies and others, are not associated with severe EBV. So not to say that all of these immune arms are useless to fight EBV, but it indicates they are redundant. Whereas now, what is absolutely critical? That's the topic I want to discuss. What is absolutely critical in the way our immune system controls EBV? So let's go phenotype by phenotype. I mentioned first the B lymphoproliferative disorder and or B-cell lymphomas. So we know, all of us know that primary immunodeficiencies associated with a broad defect in T-cell immunity is associated with such B lymphocyte proliferation. Okay, so it tells us that T cells are important, but that we know for many, many years. But more specifically, so this is just, an example of such a EBV-driven B-cell proliferation in the context of a primary T-cell immunodeficiency. You see the infiltrating B cells in the lymph node. You see on the lower left side panel that there are few T cells. That is not expected in the context of EBV infection. And there are many EBV-infected cells here stained by the presence of the EBV RNA of the virus. Okay, more interestingly, more telling are the immunodeficiencies, the monogenic diseases that are associated with mostly selective vulnerability to EBV. So patients, and I will illustrate it with a case of, in a second, with CD70 deficiency. So for instance, patients with CD27, CD70, or the other molecules mentioned here, they are basically susceptible to EBV, but not to other viruses, not to fungi, not to bacteria. So there is something selective and specific for EBV. So just an illustration in one case, but there are many other cases that have been published of CD70 deficiency. So this is this boy shown on this consanguineous family. His clinical history was a stormy B-cell proliferation these B cells were EBV positive. Each time he received rituximab, it went down, but then in relapse immediately, and eventually he was transplanted and cured by the transplantation. So he had almost all of the time a very high EBV load in his plasma. So in his boy, an homozygous mutation of CD70 was found. So this is a stop codon, and it led to a loss of function mutation, and shown on this panel here, there is defective expression of CD70, which is expressed, as you will see, mostly by activated B cells. Interestingly enough, CD27 deficiency, CD27 being the receptor for CD70, is a phenocopy of CD70, and the same with ITK, which is a kinase, and there is good reasons to believe, although this is not formally proven, that ITK is involved in signaling downstream of CD27. In a context of an immunocompetent person who is infected by EBV, this is the tonsil of someone who got a tonsil removal in the context of infectious mononucleosis. So it doesn't help him, but it helps us to understand a little bit what's going on physiologically. So you see that there is expression of CD70. These are B cells. These are EBV-expressed B cells. And on the same lymph node, you can find CD27 positive T cells likely interacting with this CD70 positive EBV-infected B cells. So what's going on if CD70 is not present at the surface of B cells? Well, one can try to reproduce what is going on in vivo in vitro by looking for in vitro expansion of EBV-specific T cells when the T cells are activated by B cells expressing or not CD70. As you can see on the control on the left-hand side, within 10 days or so, you can observe the expansion of EBV-specific T cells. That does not occur if the B cells do not express, 3 examples here, do not express CD70. So clearly CD70 knockdown or mutation impairs EBV-specific T cell expansion, and by the way, also differentiation into cytotoxic T cells triggered by EBV-positive B cells. So a summary of what I've shown briefly is that in a context of CD8 T cells able to recognize EBV peptides at the surface of EBV-infected B cells, the interaction between CD77 and CD70, possibly through ITK as a mediator of signaling for T cell expansion, this is critical to control EBV. So this is now clearly established. Okay, now let's move to a second phenotype. I mentioned chronic active EBV infections. I remind you, these are situations very, very severe in terms of prognosis, where either T or NK cells are infected with EBV, and these cells are actually, in most of the cases, malignant and very difficult to control. And I will show you one example of such a patient in whom we found a deficiency in the so-called CD137 molecule, also named 41BB. So let's have a look to the case report. So this is the family, again, with consanguinity. So the boy labeled in black developed typical symptoms of chronic active EBV. So recurrent hydroa vaxiformis skin lesions shown on the top. He had recurrent fever, EBV-driven lymphoproliferative disorder with hepatosplenomegaly, massive lymph nodes enlargement. And he had very high viremia, and it was detected both in his tumors as well as in the blood, as you will see, that he had T cells infected with EBV. They are here in lesions. This is in the liver. And unfortunately, that disease in this boy was so severe that despite the treatment, he died at the age of 14. So, and this is the detection of EBV plus, so EBV-positive T cells in his blood. So it's a very useful way, actually, of diagnosing this disorder by detecting in the circulation EBV-positive T or NK or whatever cells. So you see, they were mostly in CD4, and they were monoclonal T cells, and not in B or NK cells in that instance. So in that boy, an homozygous mutation was found in the gene encoding CD137. Not going into the detail, but it led to a frameshift mutation and loss of function of CD137. So I remind you that 41BB or CD137 belongs to the TNF receptor family and binds to its ligand, I will mention later, CD137 ligand, and this 41BB molecule is highly expressed by activated T cells and NK cells, and is believed to be a co-stimulatory molecule for CD8 T cells. Okay, in this case, just to show you, there was a defective expression of the molecule on activated T cells, and this is just to show that the interaction between the trimer of the ligand with the trimer of CD137 is inducing signals through an NF-kappa B of activation of T cells. But what it is doing exactly in the context of EBV, I will discuss that in a short moment. Before that, I'm going to a third phenotype and a third case, which is smooth muscle tumor associated with EBV. So this unfortunate girl at the age of 12 years had multiple tumors in her body, shown by the arrows here in the liver. In association with these tumors, she had very high EBV load in the plasma, and in the blood, there were some, not many, but a few circulating CD8 T cells infected by EBV, so a little bit of chronic active EBV infection, if you like. But more importantly, the tumors, some of the tumors were removed and were full of smooth muscle cells expressing EBV, which is shown here in brown. These are the EBV positive cells within the smooth muscle found in many places in the body. So in this family, so the girl is mentioned with the arrow. She had an homozygous mutation in the ligand of CD137, and actually other patients have been found to have similar mutations of the same gene. And this is inducing, again, it's the same story basically, defective expression of the molecule compared to controls. And the molecules, for instance, is not expressed by the smooth muscle cells infected by EBV, whereas there are control tumors not found in the context of this mutation where the molecule is expressed. And of course, the mutation is also leading to defective expression on other cells expressing physiologically CD137 ligand like EBV-infected B cells or dendritic cells, for instance. Again, back to an immunocompetent person. This is, again, a biopsy of a tonsil at the time of EBV infection. And you have 3 examples of patients who had infectious mononucleosis. Of course, again, these are immunocompetent people here, number 1, number 2, number 3. And you can see these are the EBV-infected B cells, in red, B, in brown, the detection of EBR in the 3 instances, different magnification. And here you have the expression of CD137 ligand in red, in brown are another marker of B cells. So not all, but some of the B cells do express CD137 in a context of infectious mononucleosis, so potentially involved in an immune response between the T cells and the B cells. So the very same kind of experiment I showed you for CD70 was performed in this context. So this is here looking assessing in vitro the expansion of EBV-specific T cells, stimulated either by cells, in these 2 cases, B cells expressing the ligand of CD137. So you see after 10 days, there is quite a very significant expansion of such cells, whereas with B cells that do not express CD137, this is not observed, and by correction, by adding a normal copy of the gene in the patient B cells, this is restored. So clearly, again, as I showed you for the CD27-CD70 interaction, it looks like that the interaction between CD137, expressed by activated T cells, and its ligand, expressed among other cells by EBV-infected B cells, is also critical for T cell expansion and differentiation. And obviously, these 2 pathways are non-redundant, since any mutation in one of them is leading to severe EBV infection in a phenotype or another. Okay, but this is not the end of the story. There is, of course, that you know very well, the context of HLA and sometimes B-cell lymphomas that are associated with monogenic diseases caused by mutations in genes involved in cytotoxicity, like perforin, RAP27A, and so on. But more telling, again, is the context where HLH and or B-cell lymphomas is triggered by a mutation leading to a selective or almost selective vulnerability to EBV, and one is very well known. These are the mutations of SH2D1A, or encoding SAP, that is associated with the SLAM molecules, and that is expressed by T cells. So let's briefly recapitulate what is known. I will not show you data here. So this SAP molecule encoded by SH2D1A, mutated in XLP, is associated with molecules of the SLAM families like 2B4 and TBA, and it leads to homotypic interaction with B cells expressing ligand like NTBA or another molecule of that family, CD48, found deficient, by the way, also in HLH. So here we have another pathway that is critical for the control of EBV, but that is mostly involved there are several functions, but here probably the most critical function is to maintain and strengthen the interaction between cytotoxic T cells and B cells, so involved indirectly in favoring the killing of EBV-infected B cells. So if I summarize what I have said so far, we have now 3 pathways that are critical for the control of EBV infection: CD27, CD70, 137 and its ligand, both of them involved in permitting expansion of T cells as well as differentiation, and a third one, 2b4 CD48 mostly, through SAP induction of signal in T cells, that is important for cytotoxicity. So 3 very key pathways that are critical to control EBV, but not other infections. That is the interesting point. It's not the end, almost the end, but there is a 5th phenotype I'd like to briefly describe to you, which is severe acute, but eventually resolutive infectious mononucleosis, something intermediate between what is occurring in physiology of infection versus the very severe cases I mentioned. So this was observed and described recently by Sylvain Latour. So in a few patients, and including some people in Finland, because the mutation, one of the mutations found, I will now describe, is quite frequent in Finland. So they were children infected by EBV. They got a severe EBV infection requiring hospitalization, treatment, some persistence of EBV, but at the end, after some months or 1 or 2 years, everything resolved, and the patients are no longer patients afterwards. So what was found in the first families studied were mutations, biallelic mutations, in a gene encoding the alpha chain of the receptor for interleukin-27. And this is leading, and I will show you on the next slide, but you have to know that this receptor is expressed by activated T cells, among other cells. And here there are examples of 2 patients where the molecule is not there compared to control after activation. Okay, so I remind you that interleukin-27 is an heterodimer between P28 and EBI3. Interestingly, EBI3 is, as I will show you, is produced by B cells once infected by EBV. So it binds to an heterodimer of the alpha chain, the one that is defective in these patients, associated with GP130, and then there is signaling through STAT molecules. Okay, so again, this is the deficiency as observed here by immunoblots in patient cells, in T cells of the patients. So now we have to discuss what can be the role of interaction between interleukin-27, this heterodimer, produced in part by infected B cells, and its ligand, so the receptor expressed by activated T cells. So what can be shown, for instance, is that T cells that are defective in the receptor, they do not activate they do not phosphorylate STAT1 and SAS3 in response to IL-27. In contrast to control here, this is obvious. They do not respond, so the receptor is not working. And the same is almost the same story as the one I mentioned earlier for CD2770 and CD137. In the absence of this cytokine receptor, the expansion with some, it's not exactly the same, but basically it's almost the same. In vitro, the expansion of EBV-specific T cells is impaired, and this is clearly shown here for patient 2. It's a bit more complex for patient 1 as compared to control. So again, this pathway between the IL-27 cytokine and its receptor plays a role in the control of EBV, and this is just to show you that EBV-infected T cells do produce IL-27. It makes sense. Now the story is a bit more complicated. I will not go into the details. If you are interested, we can discuss it, why the disease is only transient and not persistent, in contrast to the other phenotypes I described. But we have another, in any case, we have another pathway that is required for the control of EBV. And now if I summarize everything I showed you, we have 4 pathways. 3 of them, CD27, 137, and 27 cytokine, binding their ligands, are required for optimal CD8 T cell, EBV-specific CD8 T cell expansion, as well as their differentiation into cytotoxic cells, plus a fourth one involved in cytotoxicity. And all four are critical. All four are absolutely required. So the question that comes from these observations is why are these pathways, and by the way, maybe others yet to be described, critical for immunity to EBV and not for immunity against other viruses? What is specific about EBV? So here we can only speculate and maybe consider that what is specific for EBV is the fact that it induces B-cell expansion or other cell expansion, whereas other viral infection, if you think of HSV, flu, CMV, whatever, varicella zoster, they infect cells, but they don't transform them. So at the time of EBV infection, there is an expansion, at least, of B cells infected by EBV, and those cells have to be controlled. So it requires a massive expansion of mostly CD8 cells specific for EBV that creates the infectious mononucleosis, and then these cells disappear. There are some figures we can put on this. We know that in an adult, there is approximately 1 million naive CD8 T cells that are specific for EBV in the body. That's an estimation. And it can be calculated based on the kinetics of what's going on in an infectious mononucleosis, that within 4 to 7 days, these cells expand at least 14 times, leading to a massive number of T cells, of something like 1/10 to the 10. So it's a massive expansion with a division time of something like 6 hours. So we may propose that in order to get such a massive expansion and differentiation of CD8 T cells, you need many co-activation molecules. And if one of them is not there, is missing, so the one I mentioned, then the CTL growth is impaired, and the EBV infection and the transformation of B cells or of other cells is not under control. And that's for cytotoxicity in the context of SAP deficiency. This is the same story. So that's the idea that what is specific about EBV. And now if you look at these pathways, we know that for SARS-CoV-2, okay, if you have a defect in the T-cell receptor signaling, you may have a severe SARS-CoV-2 infection, but none of the other genetic defects do cause severe SARS-CoV-2, so CD27, SAP, and so on. Same for flu. For CMV, it's almost the same, whereas for EBV, you need all of these molecules. So this is the specificity of EBV. So just to conclude, I think immunity to EBV is unique because of the transforming characteristics of the virus. So we can say that the study of severe EBV infection is a kind of a blueprint for the detection of T-cell inborn errors of immunity, given the importance of T-cells in the control of EBV. And now, based on what I showed you, we need to provide a more quantitative, integrated view of T-cell responses triggered by EBV. Not all of these molecules are expressed at the same time, so there is a complex kinetics in this expansion of T-cells that needs to be deciphered. And medically speaking, it could be that using, for instance, agonistic antibodies against some of these co-molecules expressed by T-cells could be used for a better control of EBV infection in the context of primary immunodeficiency or secondary immunodeficiency, as observed, for instance, in the context of organ transplantation. Okay, I'm stopping here just to mention people involved in this work, mostly Sylvain Latour, Emmanuel Martin, Damien Boutboul, Bernard Fournier, and others, with collaborating people in Finland, I mentioned, and also like to mention Geneviève de Saint-Basile, Fernando Sepulveda, Gaël Ménaché for the study of HLH. Thank you very much for your attention.
Thank you, Alain. You have been perfectly on time, so thank you very much. And thank you also for this comprehensive overview of the vulnerability of different inborn error of immunity to EBV. And there are some, please, Claudio.
Of course, excellent presentation. I have no words. I was wondering which one of the 4 pathways are associated with the isotype switch. The reason of this question is because we observed in a few cases the persistence of a IgM response and a very long delayed isotype switch without any genetic defect.
Yeah, actually, there are two things. These molecules are not critical for IgM switch to IgG, EA, and so on. It does occur when these molecules are absent. Now, the persistence, what is frequently observed in severe EBV infection of IgM and IgG anti-VCA and not the viral capsid and not against EBNA molecules, is probably secondary to the T-cell immunodeficiency where the immune system doesn't see peptides coming from the EBNA molecules. So it's probably something secondary and not immediately the consequences of such deficiencies in these molecules.
Alessandro.
Thank you, Alain, for this wonderful lecture. I was wondering, based on your last data, do you think that in cases we have patients who have suffered severe EBV-related monocloses, which resolves, do you think we should look for the genetics in these patients now?
I think, yeah, I agree. I mean, in any case, any atypical, quote-unquote, atypical EBV infection requires a genetic analysis, including those ones that are remitting after some time. Your question allows me to say that probably the remission occurs because, surprisingly enough, interleukin-27 is required for optimal expansion of EBV-infected B cells. So in the absence, there is a dual function. One is to activate the T cells, but the second one, that goes in the opposite direction, it is required for B cell expansion. Didn't bring the data, but this is clear. So perhaps this is explaining why the disease has a limited time course compared to the other molecular defects. But sure, I will recommend to test these patients because also we do not have long-term follow-up, and we can't exclude, for instance, that maybe 5-10 years later, they may again have such a severe infection. So yes, I would say, I would recommend such analysis.
Paganelli.
Thank you very much for this important contribution. I want to ask you whether some of your findings may have relevance to the etiology of multiple sclerosis, or at least EBV involvement.
The proper answer is I don't know. What I understand from the study, we are not working on multiple sclerosis, at least up to now. There are two hypotheses, maybe not excluding one or the other. One is that there is cross-reactivity between antibodies to EBV and some glial molecules. But the second one, which I have to say, is that EBV is triggering B-cell expansion, including B cells that are autoimmune and producing autoantibodies that may play a role in the context of multiple sclerosis. And by the way, it's also possible that such B cells do better present antigens, so autoantigens to T cells that are infiltrating the nervous system. Maybe other things too, I don't know. Now, the relationship between what we know about the effectors of immunity against EBV, up to now, I don't see the link, but perhaps one day there will be one.
Alain, can I ask you one? You know, and if I'm not wrong, CD21 is the receptor of EBV, letting him to get into the B cells. So, and there are some primary immunodeficiencies which are characterized by the absence of CD21. So are these immune deficiency protective against EBV infection?
I don't know because we have never studied such a case, but I would guess yes. Yeah. Also, one should be careful because, and cautious because infections of other cells like smooth muscle cells, epithelial cells, T cells, NK cells is not CD21 mediated. By the way, we do not know exactly how EBV enters such cells. So perhaps this is not a full protection not to express CD21.
Okay, thank you. And
Bene, possiamo muoverci verso il secondo relatore, il professor Raffaele De